Thanks for the invitation. Thanks to Marco, Franca, and it's nice to be here again after, I think, 4 years. It was in four, right? 2011, the other autumn school. It's always a pleasure to be back in Varese. Actually, it's a place that I visit quite often for many reasons. One of these is the excellent collaboration that we have here with our friends, with Marco's group. And I'm not going to talk for 2 hours, so relax. So I'll take my time. I have a nice number of slides, but and so we will have time for discussion, if you will. The idea is to, as you can see by reading the title, is to give you a general overview of the role that neuroinflammation and proteotoxicity. And when we talk about proteotoxicity, we refer obviously to alpha-synuclein, the role that these two phenomena play in the pathogenesis of Parkinson's disease, a sort of crossroad, crucial to the for triggering and pushing forward the neurodegenerative process, which is at the base of this neurological disorder. So I will give you a general overview, a few data from our laboratory, but most of all, I would like to give you the general picture, if it's possible. So I will start with an overview of the clinical epidemiological features of the disease. Then we will talk about the pathophysiology of Parkinson's disease. So what goes wrong and where this, whatever it happens goes wrong. Pathogenesis of Parkinson's disease, we will enter into the focus issue of my talk. So the role of alpha-synuclein-mediated proteotoxicity. Then we will move to the role of neuroinflammation, and then we'll try to establish, to discuss briefly the connections that have been recognized that link these two phenomena, which are intimately connected. So let's start with the Parkinson's disease clinical and epidemiological features, because I think it is a nice thing to do to put the whole the disease into the right perspective. So before going into detail into mechanism of the disease, let's see, first of all, what the disease is about in terms of clinical manifestations. As you know, Parkinson's disease is the prototype of movement disorders. There is a number of typical motor symptoms: tremor, rigidity, bradykinesia, postural instability, so the tendency to fall quite frequently, that affect these patients. So PD equals to a problem in the execution of voluntary movements. But Parkinson's disease is not just about movement. That was the next slide, actually. It is a disease, and the same thing applies to Alzheimer's disease, for example, that increases with age. So it is a disease that it is unrecognized, let's say, until the fifth decade of life. And then the prevalence of the disease starts to increase dramatically. So we have, let's say, after 6 years of age, between 1 and 2% of general population that starts to develop, to manifest the disease, and this percentage increases, reaching around 5% in people over 85. I was saying that it is not just a motor disorders, because, and this has been quite recognized by clinicians, the clinical constellation of Parkinsonian patients includes also a number of very important non-motor symptoms. Non-motor symptoms that now have been recognized as a core component of the disease. Non-motor symptoms that can affect various compartments. We have autonomic dysfunctions, for example, gastrointestinal dysfunctions. More importantly, as you may know, constipation is a very frequent occurrence in these patients, and it is a symptom that further worsen the quality of life of Parkinsonian patients, in addition to all the movement problems, and then genitourinary dysfunctions, cardiovascular dysfunction, orthostatic hypotension, for example. So the problem with blood pressure is another very frequent non-motor symptom in these patients. Cognitive dysfunctions, these were thought to be a late finding in these patients, but instead it has become quite obvious that it is a problem which is present probably since the beginning. Only we need the right methodologies to detect it. Sleep disorders, particularly disorders of the REM phase of sleep. This is another very frequent non-motor manifestation. Mood disorders, depression, which affects with a high frequency Parkinsonian patients. Pain, and then non-motor symptoms that are related not just to the disease but to the pharmacological treatment. For example, the impulse control disorder, which is related to the use or abuse of dopamine agonists, psychosis, and dyskinesias, for example. Some of these symptoms may precede the onset of motor manifestation even by many years, which is why some of these symptoms are currently under investigation as potential pre-symptomatic markers of the disease. Constipation, for example, the RBD, so the REM behavior disorders, even the olfactory dysfunctions. And it is, this is the schematic representation of the geographical distribution of Parkinson's disease. You can see here, so there are a couple of potential explanations for these various hypotheses, ethnic items, issues, or dietary, for example, also influences. Just to mention the first that comes to my mind, the use of curcuminoids, for example, in India, which is a class of compounds or dietary elements with a very well-known antioxidant properties, or other dietary elements, or maybe just an epidemiological bias. So the fact that probably in some countries data are collected in a more thorough and comprehensive way. But there is no clear explanation, but this is basically the situation when we look at the geographical prevalence of Parkinson's disease. PD is a progressive disease. Once it starts, it doesn't stop. It keeps progressing, and it gets more and more complicated with time. So we may think of a preclinical or prodromal phase during which possibly the non-motor symptoms may be already there. Constipation, for example. Obviously, this does not mean that all the people who suffer from constipation will develop Parkinson's disease, obviously, but it's just a statistical finding. Then the disease starts to manifest in terms of motor symptoms. It starts unilaterally, so there's one side of the body which becomes affected, and then the motor involvement affects the contralateral side, and then with the progression of the disease gets more and more complicated. Motor complications and dyskinesia, these are related mostly to the pharmacological treatment, so it is a side effect of the dopaminergic treatment. Instability and faults, psychosis, so then the psychiatric and like this. Too much. I know. My voice is going to fade away in a few minutes, so I was trying to avoid this. Okay. So it is complicated. It is complicated. It gets more and more complicated, as the clinician know. With time, it is complicated because motor symptoms are mixed with non-motor symptoms. So it is a complex situation. It is complex not just from the clinical point of view, but also in many respects. It is a sporadic, substantially it is a sporadic disease, and same thing applies to Alzheimer's disease, for example, or ALS, except for percentage 10 to 15% of cases which recognize a genetic origin, a Mendelian origin. The story about the genetics in Parkinson's disease is quite recent. It started in 1997 when it was discovered that alpha-synuclein, the gene encoding for alpha-synuclein, the queen, we might say the queen protein for Parkinson's disease was mutated. So we had the first group of genetic PD termed PARK1, and then others followed, and you can see them here. This is just a list of the monogenic forms of Parkinson's disease. They are not all, they do not have all the same importance to the clinician, of course. There is a subgroup of familial PD forms which are more important, more frequent in the population, so they have they are more clinically relevant, and these are represented here in this box. So PARK1 and PARK4, we are referring to synuclein. In the first case, we have mutations. In the second case, PARK4, we have duplication or triplication of the gene, so the gene is not mutated in a PARK4 form of genetic Parkinson's disease. PARK2 is, and this one, although it was the first genetic form of PD which was described in the literature, is quite rare in the population. On the other hand, PARK2 and the mutation affect the gene encoding for Parkin. It is the most frequent recessive form, and so this is important for clinicians. PARK6 encodes for PINK1, which is a kinase. PARK7, DJ1 is also autosomic recessive genetic PD forms. And then PARK8, where the gene involved is LRRK2, which is a kinase, and this one is the most frequent dominant form of genetic PD. So these two are probably the form of genetic PD which are the most relevant to the clinician. Another important thing and quite recent concept is that genetic factors may also have a role in sporadic Parkinson's disease. So what I showed you in the previous slide was about the monogenic, the Mendelian forms of Parkinson's disease. But in recent years, mostly the GWAS, the genome-wide studies, have shown that genetic factors may also play a role in sporadic Parkinson's disease. So polymorphic variants, for example, SNPs in the genes encoded for synuclein, again, tau, LRRK2, may increase the risk of people to develop sporadic Parkinson's disease. And one genetic factor, in particular, loss of function mutations, heterozygous mutation in this gene, GBA, which encodes for an enzyme called glucocerebrosidase. This, in particular, is emerging a very important susceptibility factor for Parkinson's disease. This one is currently considered GBA. Mutations, homozygous mutations for GBA gives you Gaucher disease, so a lysosomal storage disease. Subjects with heterozygous mutations in this gene, they do not develop Gaucher disease, but they have a far more increased risk of developing the sporadic form of Parkinson's disease. This one, the heterozygous mutation in the GBA gene, is currently considered a major vulnerability factor, or possibly the most important vulnerability factor, risk factor for sporadic PD. And we will get back to do this. In fact, GBA mutations increase the risk by 20 times, which is quite something. And if you run GBA testing in sporadic PD patients, you will find out that between 5 and 10% of all patients with Parkinson's disease, they carry this mutation, this heterozygous mutation in the GBA gene, GBA1, to be precise. Okay. So pathophysiology. So what goes wrong in Parkinson's disease, and where does these wrong things happen? Parkinson's disease is a prototype of movement disorders on one side, and the neural correlate, of course, of this movement disorder is the involvement of basal ganglia. I've been showing this for years. I'm very proud of this cartoon, so I will give you the time to enjoy this masterpiece of animation. Basically, I'm not going into the details of the scheme. This is just to give you an idea of how the system works. Basically, this is the basal ganglia circuit with all the various components. The striatum, you can think of the striatum as a sort of funnel, which is getting all the sensory motor inputs coming from the cortex, which are required to start the movement, and is conveying these inputs through the circuit. Basically, so this is the substantia nigra pars compacta, which keeps the striatum under control through the dopaminergic efferents, which impacts on subpopulations of striatal neurons expressing D1 or D2 receptors. But anyway, the idea is that then this input is processed in the circuit. It is released out of the circuit through these nuclei, which are called output nuclei, the medial globus pallidus or internal globus pallidus and substantia nigra pars reticulata, which sends out the result of this elaboration to the motor thalamus. The motor thalamus, everything is right. So if we are in normal condition, the motor thalamus sends a feedback signal to the cortex, and so we can move. In Parkinson's disease, the main problem is here. You see that. I will show you again, so you don't miss it. Hollywood style. So the substantia nigra pars compacta degenerates. So we lose dopaminergic neurons, and so the striatum is deprived of dopamine. And this basically causes a complex cascade of events in the circuit. The main consequence of this cascade of events is that the output which comes out of this nuclei is increased. These nuclei are GABAergic, so they are inhibitory. Once this output signal is so pathologically enhanced, this over-inhibition basically switch off or reduces the activity of the motor thalamus, and the consequence, this is the theory of course, is that the feedback with the motor cortex is far less efficient, and so we have the onset of the motor symptoms. Of course, it is required a certain amount of cell loss in the nigra for the motor symptoms to occur. The idea is that it is at least required between 70 and 80% loss of dopaminergic neurons before the PD symptoms, motor symptoms start to occur. Okay. As you know, the treatment of Parkinson's disease, we can treat Parkinsonian patients to a certain extent, as you know. This is a good news. The therapy is basically substitutive. So dopamine is missing. We replace dopamine, not directly with dopamine because it doesn't cross the blood-brain barrier, but with levodopa, the direct precursor, which is taken out by surviving neurons, transformed into dopamine, incorporated in the vesicle, then released on demand, which means that this is a therapy which is only symptomatic, so it impacts only on symptoms. It does not modify in any way the progression of the disease. some years ago, probably some of you remember that some studies started to say that levodopa could be toxic to the dopaminergic neurons because it undergoes oxidation, levodopa per se or dopamine. But then, and I remember a very nice paper by Marco on this specific issue, showing that dopamine lymphocyte is supposed to increasing concentrations of dopamine. Actually, when dopamine levels were very low, dopamine was indeed inducing some protection. And then, of course, exceeding a certain concentration, then the apoptotic cascade was triggered. Anyway, so it is a symptomatic treatment that does not modify the progression of the disease, and which works very well for the first 5, 6, let's say 10 years. But then long-term side effects start to show in these patients. Reduced efficiency. First of all, the wearing off phenomenon. So the levodopa treatment starts to lose its effectiveness in these patients. Motor fluctuations, on-off phenomena. So these patients switch from periods of the day in which they are perfectly functioning and periods in which basically they freeze because levodopa is no longer working. And involuntary movements, these kinesias, which are directly related to levodopa, and which, in a way, if you think of it, it's a sort of paradox. So it's a disease characterized by the loss of movement, when in the final stages ends up in a disease with an excess of involuntary movements caused by the drug. So this is basically the situation with the pharmacological therapy. Then you can add other things. You can add dopamine agonists. You can add drugs blocking some catabolic pathways of dopamine. But basically, after almost 60 years, we are still forced to give levodopa to PD patients. There is no other way to treat these patients. So degeneration of dopamine-containing neurons is one pathological hallmark of the disease. The other one, very important, as you may know, is the presence of inclusions, cytoplasmic inclusions, containing a number of proteins, but one in particular, alpha-synuclein, which we already mentioned. And these inclusions are called Lewy body, as you know. Alpha-synuclein, like I was saying, is the main protagonist of Parkinson's disease. It's a small cytosolic protein, so there are other two forms, beta and gamma, but this one is the most important, the most represented in the brain, 140 amino acids, an alpha helix structure, particularly enriched in presynaptic terminals, and we will see why this is so important, particularly for Parkinson's disease. If alpha-synuclein at a certain point, for any reason, becomes insoluble, it starts to precipitate. It starts to, so it loses the alpha helix structure. It starts to polymerize. It undergoes through a different steps of organization, monomers, soluble oligomers. Then it starts to organize in protofibrils, then fibrils, and then we have the mature Lewy body. A major breakthrough when we speak of alpha-synuclein and Lewy bodies in particular came in 2003 when this group of German pathologists described a very peculiar behavior, we might say, that affect Lewy bodies. So until that time, it was thought that Lewy bodies were basically confined to the nigrostriatal system, and that Lewy body occurrence was strictly connected to the onset of the motor manifestations. What this group proposed and then demonstrated with this paper and many others that followed is that actually Lewy bodies can occupy the whole brain, And this colonization starts in the very early phases of the disease, possibly in the pre-symptomatic phases of the disease. And it starts, this colonization by Lewy bodies starts in the lower portions of the brainstem. For example, in the dorsal motor nucleus of the vagus, which is basically more or less here. And then these Lewy bodies start to march, and they march from the lower brainstem up, and then to the cortex. Basically, according to Brack, reaching the area which we know it is the main target of the pathological process, the substantia nigra, at stage 3. So in a mid stage of this progression, which means that according to this idea, the whole, like we were saying before, speaking of the promoter, the non-motor symptoms, the whole process starts probably many years before the motor symptoms become obvious to the clinician. Okay. And Lewy bodies can also be detected outside the brain. For example, say the same group and others actually reported occurrence of Lewy bodies, for example, in the enteric nervous system, which led this group and others also to a hypothesis that the disease might also start from the periphery, from the intestine, for example, from the intestinal mucosa. So Parkinson's disease is a complex disease. It is complex in terms of clinical manifestation. It is complex in terms of pathology, and we will see it is also complex in terms of pathogenic mechanisms. So the dopaminergic defect is possibly just the tip of the iceberg, but under the surface there is much more. Okay. So now we go into the core of the presentation. So we talk about pathogenesis. Pathogenesis of Parkinson's disease, starting with the role of alpha-synuclein-mediated proteotoxicity. But first, I'd like to give you an idea of the complexity of the pathogenic puzzle of Parkinson's disease, because it is not a monofactorial disease. There are many different mechanisms that can intervene, interacting with each other. And I try to summarize these mechanisms into 4 main categories, which are quite nicely connected to each other. Mitochondrial defects, historically speaking, probably is the most important one, because the complex 1 deficiency was a major breakthrough at the end of the 80s when it was demonstrated that this enzyme was one of the main biochemical targets of the disease. And the story, as you probably remember, came after the demonstration that there was that toxin, MPTP. MPTP toxin was basically targeting this enzyme. MPTP, for those who know, and for those who doesn't remember the story, is it was a compound. It was basically a contaminant of a drug of abuse that was synthesized in I think in California. So the story is that there was this group of students, so all young people, that all of a sudden and all together started to display PD motor symptoms. And this group of neurologists, basically Bing Langston, studied this pattern of Parkinsonian patients, and basically they discovered that the PD symptoms were due to this unintentional self-administration of this MPTP, because they had asked probably a friend of a chemist, not a good chemist probably, friend of theirs, not a good student in chemistry possibly, to synthesize some meperidine, I think. And this chemist gave them instead MPTP. So they injected these compounds. MPTP, then it was discovered that crosses the blood-brain barrier. MPTP by itself is not toxic. but it crosses very easily the blood-brain barrier. Once in the brain, it is metabolized by monoamine B into MPP+. MPP+ is the actual toxin. MPP+ goes into the dopaminergic neurons because it exploits the dopamine transporter. Once in the neuron, concentrates into the mitochondria. Once in the mitochondria, it blocks complex 1. So this is just a brief story about how and why complex 1 was studied in these patients. Then other demonstration that the mitochondria are involved came later on with the evolution of the concept of mitochondria. Once mitochondria, it became obvious that mitochondria are not static organelles, but they can fuse and divide. So there is a quality control system for mitochondria. It also became obvious that PD patients or PD models, experimental models, show an impairment in the quality control, for example, also in mitophagy. Synuclein aggregation, and we will see more in detail about this. Oxidative stress. Well, oxidative stress is obvious. If we speak of mitochondrial defects, everything that affects mitochondria function increases the rate of ROS formation, so increases the oxidative stress. And then neuroinflammation, with the main actor, not the only one, but probably the main actor, microglia activation. So these are the two mechanisms that we will discuss a little bit, starting with alpha-synuclein aggregation. So this is a review by a group led by Dino Di Monte. He works in Bonn. He's one of the most important researchers in the field of synuclein aggregation and spreading. And I like to show this slide and the following one because it gives you the idea of how our understanding of Parkinson's disease has changed when synuclein entered the stage. Because this is the year when James Parkinson described for the first time Parkinson's disease. Lewy bodies were described for the first time in 1912. In the early 60s, levodopa, the major breakthrough in the therapy of Parkinson's disease. And then in 1997, Maria Grazia Spillantini demonstrated that Lewy bodies are basically made of alpha-synuclein, and then a new era starts. So we might say that we had an old pre-alpha-synuclein Parkinson's disease, and now we have a new post-alpha-synuclein Parkinson's disease. The old Parkinson's disease was nigrostriatal centric, so everything was revolving around the nigrostriatal system. The new Parkinson's disease is a whole brain, possibly a whole body disease. Think of the Lewy bodies have been described, for example, in the intestine. So can you still think that Parkinson's disease is just about the nigra? Maybe it's not just about the brain. It's possibly about the whole body. Clinical features. All Parkinson's disease was all about motor impairment. The new Parkinson's disease is a complex clinical syndrome with motor and non-motor manifestation. We already have seen this. Mechanism. All Parkinson's disease was just about dopamine toxicity and dopaminergic cell vulnerability. The new Parkinson's disease, synuclein is the factor that is driving the whole story, and so on. I tried to summarize, of course, after 1997, there was an overflow of publications on synuclein and proteotoxicity in Parkinson's disease. So I tried to somehow to summarize the two major concepts that, in my opinion, possibly not just in my opinion, have emerged as the most important one of the past 10 years. The first was that pathological alpha-synuclein can transfer from one cell to another. And the other one is that it is possibly the soluble form of alpha-synuclein, so alpha-synuclein oligomers that are the actual toxic species, so the the form that is toxic to neurons or to synapses. Probably not the aggregated form of alpha-synuclein once it is conglomerated inside the Levy body. So this is a soluble form of alpha-synuclein. And speaking of the cell-to-cell transfer of pathological alpha-synuclein, the story begins in 2008. So it's a quite recent story. And it begins when Nature Medicine publishes two papers from two independent groups in which the authors studied the brains of patients who had been who had received 10, 15 years before a transplantation of fetal neurons. There was a time between the end of the 80s and the beginning of the 90s in which there were various attempts of surgical treatment for Parkinson's disease with fetal transplantation. So not stem cells, but fetal cells, so cells that were already differentiated into dopaminergic neurons. Some of these patients with time passed away and brains were collected, but basically the incredible discovery was that dopaminergic cells had been transplanted into the striatum. Okay. Well, Levy bodies, alpha-synuclein positive Levy bodies were found in the transplanted neurons. So, to say in a probably incorrect way, but it gives you an idea, it is as if the transplanted neurons had been infected with the Levy bodies. Okay. And this changed everything, because just less than 1 year later, it was demonstrated that alpha-synuclein can be transmitted to neighboring neurons, both in vitro and in vivo, producing Levy body-like inclusions. And with time, various mechanisms have been proposed that may mediate the cell-to-cell transfer of alpha-synuclein. Various mechanisms that includes, for example, the release of exosome cargos carrying alpha-synuclein inside and transferring from the donor cell to the acceptor cell, exocytosis mediated transfer of synuclein, formation of tunneling nanotubes, although this one is probably less recognized as a probable mechanism. Exosome probably is now considered the most important way of that synuclein may use to travel from one cell to another. And these, of course, trigger the main thinkers of this of the PD field to propose this sort of equivalence. So Parkinson's disease might be, or synucleinopathies in general, might be a little bit like prion diseases, or maybe it is a prion disease. There is a lot of debate on this, of course, and nobody knows the answer. But it is quite suggestive if you think of the way the synuclein has been demonstrated to travel from one cell to another, thinking that the prion proteins is doing mostly the same thing. This hypothesis might not be as crazy as he was sounding 5 or 4 years ago. This is I just picked two experimental papers demonstrating that this is actually true, and that the cell-to-cell transfer and travel of alpha-synuclein can occur in animal models of Parkinson's disease in particular. This is a quite famous study that was published in Science 3 years ago by the group of John Trojanowski and Virginia Lee, and in which they basically demonstrated that the intrastriatal inoculation of this should be an alpha here, of synthetic synuclein fibrils leads to cell-to-cell transmission of pathologic alpha-synuclein and the formation of Levy bodies in the substantia nigra pars compacta. And this transfer is accompanied by cell loss in the substantia nigra. We have to remember that the striatum, it is where we have the terminals of the dopaminergic neurons, but the cell body stays in the substantia nigra pars compacta, which is far, far away from the striatum. It is a long journey to make for these inclusions. So this is just a few pictures of what they found. And so basically they inoculated these preformed fibrils of alpha-synuclein, and this inoculation seeded the aggregation of endogenous synuclein in wild-type mice. And this seeding process was obviously increasing with time. And it was colonizing basically the whole brain. But most importantly, this seeding was involving the substantia nigra pars compacta, where the cell bodies are residing, and was associated with a progressive cell loss. This is another study by the group of De Monte in Bonn. The experimental approach was slightly different. In this case, they injected adeno-associated viral vectors carrying the synuclein gene. So they induced the overexpression of alpha-synuclein in the vagus nerve in the neck of these rats. They injected in the neck, let's say, at this level, and then they studied the eventual spreading of alpha-synuclein throughout the vagal nerve and then the brainstem. And this is actually what they saw. So they saw that there was an accumulation of alpha-synuclein starting from the injection site and then proceeding through the brainstem and proceeding more and more as time was passing, until basically they found synuclein inclusions in the mesencephalon, so at the level of the substantia nigra pars compacta. So synuclein can travel. But why is it toxic? Because one of the misunderstandings that is still around somehow is that synuclein per se is toxic. Synuclein per se is not toxic. Synuclein is just a protein. It is a protein with a number of physiological functions that are being discovered every day. It is the pathologic synuclein, so synuclein starts to precipitate that it is toxic. And the toxicity of synuclein is mostly related to the oligomeric forms of alpha-synuclein, as we said before. So if we want to, of course, if we want to understand why synuclein is toxic, first of all, we have to understand what synuclein is doing physiologically speaking. So we said that it's a small protein abundant in the cytoplasm and the presynaptic terminals. What we know about synuclein is that it is for sure involved in the dopaminergic transmission. It is involved in the synthesis, so it interferes, for example, with the activity of the tyrosine hydroxylase. It reduces the activity of TH, which is one of the enzymes involved in the synthesis of dopamine. It is involved, most importantly, in the release and reuptake of dopamine. Why? Because alpha-synuclein interacts at the presynaptic terminals with this NER complex. NER complex is a multi-protein complex, which is basically the core machinery required for the vesicle fusion with the plasma membrane, the release of transmitter into synaptic cleft, and then reuptake of the transmitter. So this is certainly one of the things that alpha-synuclein does, which makes sense. I mean, if alpha-synuclein is involved in the dopaminergic transmission, if alpha-synuclein does not work well, dopaminergic transmission is not working well. So all makes sense. The problem in Parkinson's disease is that synuclein oligomers they probably impair SNARE function. So basically they derange the system, so they basically impact negatively on the efficiency of the snare complex in terms of vesicle fusion and release of transmitters into the synaptic cleft, which basically leads to reduced dopamine release. And so this is a functional consequence. Increase cytosolic dopamine, right, because dopamine is no longer released efficiently, so it starts to accumulate in the cytosol. But dopamine undergoes autooxidation. And autooxidation with increased release formation of reactive oxygen species leads to synuclein oligomerization. So this is a situation that probably we are facing when we reason on the effects of synuclein oligomers within the dopaminergic synapse. Another target of synuclein oligomer toxicity are certainly the mitochondria. Mitochondria, we go back to the beginning of the story in PD pathogenesis. So, I mean, mitochondria involved in the PD pathogenesis since the very first moment. Synuclein is confirming that mitochondria are a target, a preferential target of the disease, because under normal conditions, most of synuclein is cytosolic, so it does not associate in mitochondria except for a small fraction. The things change radically when synuclein starts to precipitate, because oligomeric synuclein instead may interact quite heavily with mitochondria, causing mitochondrial defects, complex 1 inhibition, again, damage to mitochondrial DNA, increased mitochondrial fragmentation. Another interesting finding, quite recent, is that synuclein probably resides at a specific intracellular level, which is this MAM, mitochondria-associated membrane, which is a physical domain that puts into physical contact the endoplasmic reticulum with mitochondria. It's here. So one of the most recent ideas is that this is the house of alpha-synuclein. Pathological alpha-synuclein loses its association, and this may alter the endoplasmic reticulum mitochondria communication with a cascade of consequences you can imagine. So trying to summarize what we have seen so far, pathological alpha-synuclein impairs snare function, and this causes a reduction in the release of dopamine, which leads to increased cytosolic levels of dopamine, which undergoes autooxidation, so increased oxidative stress. On the other hand, we have seen that pathological alpha-synuclein can impact on mitochondrial function, causes complex 1 inhibition, damages the mitochondrial DNA, causes increased mitochondrial fission, and this again gives you oxidative stress. So we can think of alpha-synuclein different with the scheme that I showed you before, the puzzle. Probably it is not a real puzzle. We can think probably of a scheme like this, sort of bullseye, with alpha-synuclein aggregation in the center, driving oxidative stress, mitochondrial defects, but there is one piece missing, and the other one is this one. The other thing that the alpha-synuclein, pathological alpha-synuclein does very well is activating the microglia, so triggering neuroinflammation, and this may complete the story. Maybe I will talk 2 hours. No. Okay, so this brings us to the role of neuroinflammation in Parkinson's disease pathogenesis. neuroinflammation is I know that this sounds like a joke, but it's a hot topic in Parkinson's disease. There's been tons of publications in the past 10, 15 years about the role of neuroinflammation in this disease. Just a few basic concepts that we might think of when we approach neuroinflammation. I'm not an immunologist, but I think that a few concepts should be shared by everyone who's involved, who wants to study the pathogenesis of Parkinson's disease, of neurodegenerative diseases. The brain is not an immune privileged system as we once thought. It is capable, in fact, as we know now, of dynamic immune inflammatory responses. These responses are mainly mediated by the activation of microglia, which is, as you know, the macrophage resident population in the central nervous system, and also by astrocyte reactivity. There is a communication between the central nervous system and the peripheral immune system. Immune cells can can pass from the periphery to the brain and vice versa, and this is a rather common phenomenon. And as you probably know, a big support to this concept was given just a couple of months ago by this paper published in Nature. And Kipnis is a guy who knows what he's talking about when he speaks about neuroinflammation. In this paper, for the first time, these authors described a system of functional lymphatic vessels lining the dural sinuses, which is something that had been never demonstrated before. So lymphatic vessels in the meninges, able to carry both fluid and immune cells from the cerebrospinal fluid. And as they state quite correctly, this is a discovery that calls for a reassessment of the basic assumptions in neuroimmunology, and might shed new light on the origin of neuroinflammatory and neurodegenerative diseases. So, keeping all this in mind, it is not a surprise to see that there is a lot of neuroinflammation markers that are modified in Parkinsonian patients. There are many studies published in the past 20 years showing that there is a plethora of cytokines which are increased, for example, in the cerebrospinal fluid, or in serum, or in brain tissues of Parkinsonian patients. This is another quite well-known study by the group of Dave Brooks, in which they studied the microglia activation with a neuroimaging procedure, basically a PET study with this ligand, the PK 10195, which binds activated microglia, and they demonstrated that activated microglia is widespread in the brain of Parkinsonian patients compared to normal healthy controls. So this tells us that microglia, as we mentioned before, is the actual effector, the actual protagonist of this story, Parkinson's disease, microglia, which can react to an infinite number of triggers of any possible kind. You think of a potential trigger, microglia will respond. And when microglia is activated, one of the things that they do, they release nitric oxide, because microglia contains lots of inducible nitric oxide synthase. So activation of microglia results in the mass information release of nitric oxide, which generates reactive nitrogen species, which basically join the party of oxidative stress and increase the oxidative damage to dopaminergic neurons. This is the idea. A microglia activation is most likely a very early phenomenon in the progression of the disease. This is a cartoon of a review by Dianish Stefan, who proposed this model, which I think might be quite reliable. So this is referred to the effect of MPTP. MPTP is also very used to replicate PD pathological phenotypic features in animals, mice or monkeys, but probably these may also apply to patients. So what the idea is that once the neurotoxic insult has been delivered, there is a phase of neuronal distress during which there is still very little neuronal damage, but during which microglia is already activated, and then it stays activated as the neuronal distress phase progresses, turning into a phase of manifest neuronal death, being joined at a certain point by T-cell infiltration and astrogliosis. Microglia is certainly a very early phenomenon in Parkinson's disease natural history. Another thing that happens, as you know, in the brain of animal models of Parkinson's disease, but most importantly in the brain of patients, is that T lymphocytes can infiltrate mesencephalic areas in these patients. But what is the connection between alpha-synuclein aggregation and neuroinflammation? I mean, pathological alpha-synuclein might be a trigger of microglial response. Probably it is, and there are many data supporting this strict connection linking alpha-synuclein pathology to neuroinflammation. There are in vitro studies, for example, showing that extracellular alpha-synuclein, because alpha-synuclein is also extracellular, of course. I mean, like we said before, it gets incorporated and starts to be packed into the living bodies. It is soluble, it goes around. So extracellular alpha-synuclein promotes the release of cytotoxic soluble factors from microglia or cell cultures using, for example, human monocytic leukemia cells. These properties, so this ability to trigger microglia activation, is even more pronounced in the presence of mutant forms of alpha-synuclein. So these are the mutant forms we have, for example, in the PARK1 form of familial PD. Also aggregated nitrated alpha-synuclein. Nitrated alpha-synuclein is another, is typically found in the cytoplasmic inclusion, can stimulate microglia activation, possibly by modifying the microglia proteome. And that was in vitro. In vivo, basically, this hypothesis has been confirmed because microglia activation is found in transgenic models, so in transgenic mice overexpressing wild-type alpha-synuclein, and this is accompanied by infiltration of B and T lymphocytes in the substantia nigra pars compacta. Microglia can also be directly activated by alpha-synuclein, which possibly alters the microglial expression of toll-like receptor. So all these things just to say that pathological alpha-synuclein, one way or another, triggers the activation of microglia. One thing that could be important to keep in mind, however, is that, as you know, microglia, once it is activated, it does not go into just one direction. It can split, it can polarize toward cytotoxic M1 type of activation, or toward an M2 cytoprotective type of activation. And even in this case, the transgenic models of Parkinson's disease using alpha-synuclein have been quite helpful in providing some very important insights into the correlation between synuclein and microglial activation. Just very briefly, because I'm sure that you all know what M1 and M2 mean, but just to refresh everybody's memories, which is always a good thing. Microglia, like peripheral macrophages can be activated can be polarized, like we were saying, toward a cytotoxic M1, which is a classical pathway, or a cytoprotective M2 alternative pathway phenotype. M1 phenotype means that it is triggered by pro-inflammatory cytokines or LPS, and this means that microglia starts to release a lot of pro-inflammatory cytokines, reactive oxygen species, nitric oxide, and reactive nitrogen species. This basically gives you a lot of cytotoxicity, and the meaning would be just the elimination of the threat. On the other hand, the M2 phenotype, which is triggered by anti-inflammatory cytokines, pushes the microglia towards the release of anti-inflammatory factors, because as you know, there are lots of cytokines that act as anti-inflammatory rather than pro-inflammatory agents, dampening the immune responses, promoting tissue remodeling and repair. This would be the meaning of the M2 response. An interesting thing that has been shown is that the microglial activation and polarization may be influenced by the extent of the neuronal death associated with the alpha-synuclein alteration, which means quite simply that if we have a transgenic model in which the synuclein is overexpressed at low levels, which are sufficient to induce a certain level of microglial activation, but do not trigger Niagara cell death, we will have a fast, transient increase in microglial cell numbers, which, however, will not show phagocytic features. On the other hand, if we overexpress human alpha-synuclein at a level which is actually causing cell death in the substantia nigra, pars compacta, which, by the way, is not that easy, because usually you do not have that in the usual transgenic model. You will need to inject the gene directly into the nigra using a viral vector. In this case, you will have a delayed increase in microglial cell numbers with a long-lasting expression of phagocytic macrophage-like morphology in the activated microglia, which possibly means that we might have a multimodal, at least bimodal, response at the level of microglia, which accompanies neuronal damage associated with alpha-synuclein deposition. So we might have a balance between neuroprotective and neurotoxic mechanisms interacting with each other in a complex combination, which may be disrupted in the long run if the neurotoxic insult is prolonged for enough time, finally leading to the cell death. Yeah, this is just to mention, I thought it might be of interest to some of you, since we, speaking of neuroinflammation and the connection between alpha-synuclein inflammation, just to know that there are some attempts in terms of finding of novel therapeutic strategies, trying to interfere not just with the deposition of synuclein within the neurons, but to interfere possibly with the transfer of the extracellular synuclein from one cell to another. So the revelation that alpha-synuclein is present outside cells has triggered the interest, the possibility that anti-synuclein, antibody-based therapies might be at least an attempt worth trying. So I'm not going to read through all these studies. These are very recent review by the Patrick Brundin, basically, about new immunotherapy in Parkinson's disease, collecting the data on the experimental studies with synuclein active immunization or passive immunization. The general idea is that basically in all these studies, the two different techniques induced reduced synuclein pathology and improved the behavioral outcome in transgenic models of Parkinson patients in Parkinson's disease. But as you know, I mean, if you work with animals, you know that in most cases your experiment will work in your animals. The problem is that you have to translate the experimental findings into the clinical setting. Then things change. As for the human studies, there are clinical trials that are currently ongoing. A few reports basically collected at conferences or in media showing that passive immunization, so antibodies, anti-alpha-synuclein are at least safe and tolerated as a therapeutic intervention, and that this infusion can reduce the free serum levels of alpha-synuclein in the patients that have been inoculated. But as far as I know, there are still no clinical data, data on the clinical efficacy of these procedures. But okay, I think it is something that is important to know. This is ongoing, of course. I will drink some water so we can make the 2 hours. Okay, so we have seen what synuclein does, or what we think the synuclein does physiologically speaking. We've seen what it does when it is altered, so when it starts to aggregate. But why is this happening? So are there some conditions that can trigger, that can favor the phenomenon of alpha-synuclein accumulation or aggregation in Parkinson's disease? And I summarize these conditions under 3 categories. The first one is gene mutations or duplication, triplication, so the Park 1 or Park 4 forms of familial PD, for example. Mutant alpha-synuclein precipitates far more easily than wild-type synuclein, and in the presence of duplication or triplications of the gene, there is simply too much synuclein to the cells for the health for the cells to handle correctly. Post-translational modifications, all these are quite obvious, right? After all, we've seen oxidation, phosphorylation, nitrosylation, all the conditions that we have seen so far, basically mitochondrial defects, microglial activation, are all conditions, obviously, that through the release of reactive oxygen species or nitrogen species can impact post-translationally on alpha-synuclein. But the third mechanism is the one that I'm most interested in, and the one I would like to spend a few words on. So the defects in the degradation pathways. So not qualitative problems, but let's say quantitative defects. So degradation pathways, which basically are the autophagy machinery and the ubiquitin proteasome system, which, if defected, can lead obviously to a reduced removal of alpha-synuclein, which leads to accumulation of alpha-synuclein. And what we know nowadays is that when the cell has to deal with the alpha-synuclein, the two systems are likely to cooperate. So the ubiquitin proteasome system and the autophagy machinery, they can both degrade soluble alpha-synuclein. And when we speak of alpha-synuclein in terms of autophagy, we have to remember that we have macroautophagy, microautophagy, and chaperone-mediated autophagy. Chaperone-mediated autophagy, CMA, is the one which is mostly involved when the cell has to get rid of alpha-synuclein. So until we have just soluble alpha-synuclein, the UBS, the proteasome, and autophagy work together. When things get rough, I mean, when alpha-synuclein starts to aggregate, then CMA intervenes and becomes the most important mechanism to eliminate the pathological alpha-synuclein. So these are two systems that are coordinated, that plays complementary roles, which tend to compensate each other's deficiencies. So, right, both systems can handle alpha-synuclein in physiological conditions. Pathological alpha-synuclein, on the other hand, is mostly degraded by CMA. Okay. These two systems have been shown to be implicated in Parkinson's disease. There are a number of studies showing in vitro or in vivo studies, also in patient studies, showing that UPS and autophagy machinery can be defective in Parkinsonian patients. One aspect that we are quite interested in is the investigation of these systems at the cellular level in peripheral cell of Parkinsonian patients, which is a way, we think, to try and understand a few things about the defect of these systems, because if we assume that these systems are basically in all cells, not just in neurons, with the right techniques, we might be able to see a reflex, a shadow of these defects also in peripheral cells, which are obviously much more easy to study than a neuron. For example, fibroblasts. We've been doing this for some years now. Fibroblasts are more and more used in Parkinson's disease research. We have found last year we published this study, and these are the experiments that Giulio Ambrosi did in a couple of years ago. We found that actually the activity of the ubiquitin proteasome system is decreased in the fibroblasts of Parkinsonian patients, sporadic Parkinsonian patients. And this was somehow confirming another finding that we had demonstrated some 8 years before, that the same thing, so a reduction in the activity of 20S proteasome, 20S the catalytic component of the UPA. So it's the one who's doing the work, it's the activity which is actually digesting the ubiquitinated proteins. So we had found that in lymphocytes, lymphomonocytes from PD patients, this protein 20S activity was reduced. We found the same was happening in fibroblasts of Parkinsonian patients. This reduction is associated in Parkinsonian patients with an increase in polyubiquitinated proteins, which I think is quite obvious. I mean, if the proteasome is not working, what you have is an accumulation of the ubiquitinated proteins that the proteasome is supposed to degrade, and also an increase in Parkin. Parkin is among the various things that Parkin does. Parkin is also a ubiquitin ligase, so is the one which is tagging proteins with ubiquitin tails. So this might be just a compensatory increase of Parkin, or on the other hand, since Parkin is also substrate itself of proteasome 20S, must be just a result of this reduction. Then we also studied in collaboration with a group at the Erasmus in the Medical Center in Rotterdam, the mitochondrial respiration, the fibroblasts of these patients. And we found that these cells have a specific impairment in mitochondrial respiration, which becomes particularly obvious when the fibroblasts are put under stress, for example, when they are exposed to rotenone, which is a complex 1 inhibitor. Then in these conditions, fibroblasts from Parkinsonian patients displays a vulnerability in terms of mitochondria, of metabolic impairment, far more enhanced with respect to control fibroblasts. So fibroblasts from Parkinsonian patients showed impaired proteolytic activity and a reduced ability to endure metabolic challenges. That was the background of the work that we did in the next 18 months. When something new appeared, I mentioned already this in the first slides, in the field of Parkinson's disease, when we speak of proteolysis, proteotoxicity, and Parkinson's disease, which is the role of glucocerebrosidase. A new pathogenic link. As I mentioned already, heterozygous mutations in the GBA1 gene, which is encoding for this enzyme, glucocerebrosidase, are probably the most important risk factor for sporadic Parkinson's disease. So the presence of a mutation, of heterozygous mutation, homozygous mutations, I'll say again, gives you Gaucher disease, okay, which is a lysosomal storage disorder with a number of neurological and non-neurological symptoms. Heterozygous mutations increases 20 to 30 times the risk of developing Parkinson's disease. Between 5 and 10% of all Parkinsonian patients carry this mutation. And this has been confirmed at various levels in many countries. The first cohort to be studied was an English cohort, and this English group, and we are lucky enough to be collaborating with this group. They still have the biggest cohort in the world of GBA mutants. So they found this percentage of mutants in the population, but this percentage has been confirmed, for example, in another study published last year in Italy by the group of Stefano Goldboom of the Pezzolis group, and they basically confirmed the same level of prevalence of this mutation in sporadic Parkinsonian patients. So GBA1 encodes for GCase, glucocerebrosidase, which hydrolyzes glucosylceramide, a sphingolipid. Okay. The thing is that it has become apparent that there is an inverse correlation between the activity of GCase and alpha-synuclein in the cell. This example, for example, a study conducted in primary cultures of human iPS neurons, in which the loss of GCase activity compromises the degradation, lysosomal degradation. Lysosomal degradation means CMA, so chaperone-mediated autophagy. Lysosomal degradation of alpha-synuclein causing accumulation of the protein, which leads to neurotoxicity. Basically the same results have been confirmed by another study conducted in neuroblastoma cells expressing a nonsense mutation in the GBA gene, so a non-functional so cells with a non-functional GCase. Also, in this case, there was a lysosomal dysfunction, which was associated with an accumulation of insoluble alpha-synuclein aggregates. So the idea is basically, like I was saying, that there might be a reciprocal interaction between glucocerebrosidase and alpha-synuclein that basically means that if we have a reduced activity in glucocerebrosidase, we will have an increased accumulation of alpha-synuclein and vice versa. Because also alpha-synuclein, once it starts to accumulate in its pathological forms, can impact on the activity of glucocerebrosidase, possibly triggering the occurrence of a vicious cycle. More recent studies have also started to suggest that, in fact, reduced GCase may not just be restricted to subjects with mutation in the gene, but it might be a finding shared by all the sporadic Parkinsonian patients or patients with other forms of neurodegenerative disorders characterized by the position of Lewy bodies, like the Lewy body dementia. Because it has become quite obvious that GCase activity can be reduced in various brain areas of these patients. Again, sporadic PD patients without the GBA1 mutations. Even in the CSF, there is a nice study by the group of Perugia, Paolo Calabresi, and Lucilla Parnetti. They show that Parkinsonian patients show a reduction in the activity of GCase in the cerebrospinal fluid, which is associated with an increase in a reduction of total alpha-synuclein, an increase in oligomeric alpha-synuclein, which becomes an increase, really high increase in the oligomeric / total alpha-synuclein ratio in the cerebrospinal fluid. This is a report that has pushed this item even further, suggesting that actually GCase might start to decrease with age, even in normal people, in the areas that are mainly involved in Parkinson's disease, so in the putamen and the substantia nigra. So while in Parkinsonian patients, the levels of GCase in this study remain consistently low in the patients, in normal subjects, there would be an age-dependent decrease in the activity of this enzyme. So, like this article, the title of this article is telling quite explicitly, glucocerebrosidase is shaking up the synucleinopathies. Made it. The interesting thing is that, and this is one of the things that dragging the attention of various groups in this field, GCase activity can be enhanced. One pretty curious thing, observation came in 2009 when this group did a drug screening on over 1,000 FDA-approved drugs to identify small molecules that might be useful as agents for enzyme enhancement therapy. So for therapy of diseases caused by a defective enzyme. And among all these more than 1,000 compounds, one compound that showed the most promising profile was this one, ambroxol hydrochloride. It is a small molecule known for its expectorant and anti-inflammatory activity, which may function as a molecular chaperone. So it may enhance the activity of the GCase, favoring the trafficking into the lysosome, because the story is that when GCase is mutated, basically its conformation changes and the enzyme is thought to become unable to enter into the lysosome, which is where it is supposed to work. Ambroxol, probably we all had been administered ambroxol when we were kids, or when we were having a fever or cold or cough, because it's known in Italy with the brand name of mucosolvan. And since I realized that one of the topics of this autumn school, Marco, was drug repositioning, I thought that this would fit perfectly well with the concept of this course. And ambroxol has been shown to increase the lysosomal fraction and the activity of several mutant GCase variants in the fibroblasts of patients with Gaucher disease. But could be effective in Parkinson's disease? This is the true question, at least for us who are working on Parkinson's disease. This paper was published last year in Brain. Brain, you know, is a very prestigious journal of neuroscience by the group of Anthony Shapiro of the University College of London, who's basically studying and detaining, like I was saying before, the biggest cohort of GBA mutant with or without Parkinson's disease, because one of the things that they are doing is also to see whether subjects with a GBA mutation who have not developed any sign of Parkinson's disease can develop, and how, and with what kind of progression, or in response to what kind of conditions. But anyway, they found, studying the fibroblasts of subjects with Gaucher disease or Parkinsonian patients with heterozygous GBA mutation, they found, as it was obvious, that there was a huge reduction in the activity. This is the activity on controls. Okay, this is the Gaucher disease. You can see that there is a basically disappearance, so 95% reduction in the activity of GKs. In PD patients with heterozygous GBA mutation, there was a 41% decrease in the activity. But when they gave Ambroxol to the fibroblast, not to the patients, no one has done this so far in PD patients, you can see how the GK's activity was pushed up, and in the groups. It was a sort of horizontal effect of all the groups, regardless of the condition of normal, control, Gaucher disease patient or Parkinson's disease patient. So Ambroxol with this protocol increased GK's activity in all subjects, and also reduced the internal formation of free radicals. So this is when this group started to propose Ambroxol as a potential disease-modifying drug. And we got curious, of course. And so we basically investigated the GKs and other factors involved in the in proteostasis in the fibroblast of PD patients with or without mutations in the GBA gene. And we also verified the in vitro effects of Ambroxol using the same protocol that Shapiro had used, so 60 micromolars for 5 days. These are the experiments that Julia has done and has designed and conducted. And I'm not showing you all the data, just the ones that, in my opinion, are the most interesting. So we found, first of all, that GK's activity is reduced in the fibroblast of Parkinsonian patients carrying an heterozygous mutation in the GBA gene. Just the activity. Protein levels are not changed, which makes sense. When we give Ambroxol to the fibroblast in culture, you can see how both the activity and protein levels increase dramatically. Again, confirming Shapiro's findings in all the groups, regardless of the pathological or normal condition. And this is a staining, a lysosomal staining for the lysosomal mass, which also shows you graphically how the lysosomal mass is increased after incubation of fibroblasts with Ambroxol. Then we studied other factors that are involved in the functioning of GKs. Two in particular, LIMP2. LIMP2 is a receptor which basically binds GKs and transfers GKs from the endoplasmic reticulum right into the lysosome where GKs is supposed to act. And then Saposin C, which is a cofactor that increases, so fiber basically the interaction of GKs with its substrate, so glucosylceramide. In basic basal condition, we did not find changes in Saposin C levels or in the precursor of Saposin C, which is prosaposin, between the groups. But then Ambroxol increased, almost doubled the levels of Saposin C, so of this cofactor, very crucial for the functioning of GCase. It also increased, but not significantly, the level of the precursor of sabuzin C. Quite interestingly, also increased the activity of cathepsin D, which is the cleaving enzyme that cleaves prosabuzin to give you sabuzin C. And then LIMP2, so the receptor involved in translocation of GCase from the endoplasmic reticulum to the lysosome, basal condition, there were no changes. After ambroxol, there was a moderate increase in the levels of LIMP2, which interestingly was significant just in the fibroblast of patients carrying the GBA mutation. So, to summarize this last part of the presentation, in particular this data, we confirmed that the activity of this enzyme, glucocerebrosidase, is deficient in the fibroblast of Parkinsonian patients carrying a heterozygous mutation in the GBA1 gene, and that ambroxol can actually increase the activity of this enzyme. We also observed that ambroxol increases levels of sabuzin C LIMP2, so two basic cofactor agents, molecules that favor the activity of GCase and the trafficking of GCase to the lysosome. Of course, there are open questions, very important. The first one is this one. But so what we did, and what also the group Shabira did, was just an in vitro experiment. So they put the fibroblast in cultures. They gave ambroxol to the cultures and detected all these changes. But is ambroxol actually able to engage and modulate its targets following the systemic administration, with all the many variables that intervene in this case? The second open question is even more important. So once we actually increase the activity of this enzyme, are we sure that this is going to impact significantly on the clearance of alpha-synuclein in vivo? And the third open question, which possibly is even more important, is ambroxol able to impact on the symptoms and progression of Parkinson's disease patients? So these are open questions that we and others are trying to some way to answer. So 25 minutes to go. Okay, so either I read the conclusion very slowly, 25 minutes to make 2 hours, or I just go at the normal speed, and then we can discuss a little bit. The first one we can discuss. I was sure you were going to say that. Okay, just a conclusion, quite simply, and almost tautological conclusion. Proteotoxicity triggered by pathological alpha-synuclein is crucial to the pathogenesis of Parkinson's disease, which is why I put it right at the center of this sort of target, because it is able to drive every other mechanism involved in the disease, and particularly neuroinflammation. Enhancing the ability of a cell, of a neuron, to eliminate the pathological alpha-synuclein accumulation and or transfer, which is also equally important, is a very intriguing, difficult, but fascinating strategy that may protect the neurons of PD patients for alpha-synuclein toxicity and propagation of the neuronal damage. So this is probably the new frontier where PD research are one of the new frontiers where PD research are currently working. And this is just to thank the people who help me do the actual job, because Marco, you can agree with me, we do nothing. They do the job. But we find the money, which is equally important. So this is the lab crew. Julia, who already mentioned, who's present here, today, and the other people in the lab, Silvia Ceri, Cristina Ghezzi, Rosetta Greco, Giovanna Levandis, Stefania Mangione, Francesca Siari. This is the Parkinson's disease unit led by Claudio Pacchetti. These are the clinicians and who are very important to us because we work a lot on animal models, but we also work a lot on PD patients. And without the help of clinicians, it would be impossible to try and see both sides of the coin. Stefano Goldburn is the geneticist at the Parkinson Institute in Milan, who's helping us a lot with the recruitment of GBA mutant PD patients. Pier Giorgio Mastroberardino is our agent in Rotterdam at the Erasmus Medical Center. And with this, I have finished. Thank you.